So this is Unit 9, or Microbiology, where we're going to talk about viruses and... So, a virus. What exactly is a virus? You probably have heard this term many times in your life, but what exactly is a virus? And what's the difference between a virus and a bacteria? Well, a virus is anything, or a virus or a bacteria is called a pathogen. So a pathogen is basically a cell that actually attacks from the inside. So pretty much what a virus does is it uses your own DNA and it kind of attacks you from the inside out. So unfortunately, viruses cannot be treated with antibiotics. So antibiotics actually is the only thing that bacteria you can use on a bacterial infection, but viruses, unfortunately, we cannot use an antibiotic. So even though the virus may lay dormant within that cell, okay, so dormant means to go into hiding. It's kind of like when our trees actually fall, the leaves are falling. The trees aren't dead, but they're laying dormant. So the viruses can actually lay dormant in the body for a numerous amount of years and or months or a couple days, and it will come back at a later date. So again, what's a virus? So according to Google, which we love, okay, it's an effective agent that typically consists of a nucleic acid molecule. Okay, so you've heard of nucleic acid. So that's either DNA or RNA. That's in a protein coat. You've heard that before during Unit 2 with our biochemistry. And it's basically too small to be seen by a light microscope, and it's unable to, or it's only able to multiply when we're talking about living cells or inside of a host. So here's just a table for you to just kind of give you a rough estimate of some more common viral diseases in humans. So when we talk about sexually transmitted diseases, okay, we're talking about AIDS or genital warts or even genital herpes. So some childhood diseases that you've probably had a vaccine for are the measles, the mumps, chickenpox, and even German measles. Some respiratory that only affect your respiratory system is the common cold, influenza, which is basically the flu, and SARS. So some skin disease is warts, fever, blisters, and shingles. Shingles are associated if you have chickenpox. So if you have chickenpox like I do, I already have the virus, the shingle virus, already internally in me, that I can actually get shingles when I'm older. So digestive tract, did you know that diarrhea is actually due to a virus? So the nervous system, some of those diseases are rabies. So you've probably seen movies or have seen, maybe a sick animal with rabies. They don't necessarily always foam at the mouth, but that's actually due to the nervous system shutting down. So some other diseases are like smallpox, some cancers, hepatitis, mono, the kissing disease, if you will, conjunctivitis, which is actually just pink eye, and hepatitis C. So what are some characteristics? Well, viruses are pretty simple. They don't have a membrane. They don't have cytoplasm. They have no ribosomes. They don't really have a lot of other cellular components. They have no Golgi apparatus. They don't have any mitochondria. They can't really move. They don't grow. They only can reproduce if they're inside of a host. And they basically have those two parts. They have a protein coat, or they have DNA or RNA. They're extremely tiny, and most of the time we can only see them through more advanced electron microscopes. So let's go a little review about DNA. So what's DNA's shape? Well, it's a double helix. It kind of looks like a spiral staircase. Each of the sides are actually associated or held together by a weak hydrogen bond. So remember, hydrogen bonds are extremely weak, and we actually can rip DNA pretty easily apart. So they have these repeated units of nucleotides. Do you remember what the three parts of a nucleotide is? Well, it's a sugar, a base, and a phosphate group. The latter, or basically the sides of the ladder, are the deoxyribose, which is the sugar, and the phosphate group. So in the center are those nitrogenous bases, which are A, T, G, and C. So do you remember the base pair rules? Well, adenine always goes with thymine, and guanine goes with cytosine. So if we look, here is basically an image of a more zoomed in or structurally correct of a nucleotide. So you can see the purple is the sugar, the blue G is the base, and and the yellow P is for the phosphate group. And you can see that they're connected. The guanine and the cytosine on the bottom are actually connected by that, again, that very, 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 very weak hydrogen bond. So again, structurally, okay, it's only made out of two parts. It's made out of a capsid, which is a protein coat. Sometimes they have an envelope, or basically it's kind of like an additional membrane. But the internal core, what it's made out of, is it's made out of a DNA or RNA, and sometimes they might have some proteins such as enzymes that speed up their chemical reaction. So there's a couple different kinds. There's obligate, okay? This is an obligate parasite. Basically, they can't exist without a host, and they're very, very specific to their host type. So you can say that this particular virus is only associated in humans, dogs, but there are some viruses that can actually cross species. So if you remember the bird flu, well, originally bird flu was actually found only in birds. And through human contact, humans actually were able to pick up that bird flu. So they can go over cross species. So they're very specific to the cells that they infect. So the common cold is only attacking respiratory tract. So that's why you cough or you sneeze or you sniffle, but you don't have, it's not going to attack your muscles. So HIV, again, it's very specific. It only attacks white blood cells. So this guy down here is pretty neat. This is actually called a bacteriophage, and it is actually a virus that only attacks bacteria. It's kind of neat because it kind of looks like the lunar landing spaceship. And so if you notice, it has that outside protein coat, the DNA inside. It does have a sheath that's below, and it has basically, it kind of looks like special limbs or spider limbs. It does have a core that actually injects its DNA into other bacteria. It will actually kill it. So the viral reproduction, there's two different types of cycles for viruses. There's the lytic cycle, which basically you can think about lit tick, 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 ticking time bomb. And basically when the reproduction occurs, there's just so many baby viruses that the cell actually pretty much explodes because they just cannot use, they can't, the host cell just bursts because there's so many baby viruses. So the lysogenic, okay, this is where reproduction, they don't reproduce immediately, okay? This is where these viruses lay dormant for a while, and then they eventually reproduce and will go throughout the rest of the body. So some of them can undergo both. So what we're going to do here is we're going to look at some attachment, okay, or basically how viruses replicate. So they replicate using their own genetic material as well as the host material, as well as the host DNA. So remember how I told you that some viruses, basically all viruses, kind of use your own DNA and trick your own DNA in order to create more of their DNA. So what they do is they have attachment first. They just attach themselves to the cell. They penetrate. Some of them will actually, the virus is just being engulfed by the host cell, okay? And this is where the virus can either enter lysogenic or lytic cycle. The biosynthesis is basically where, again, that viral components are made out of the protein coat, the capsid, which is the outside wall, and then the DNA or RNA, depending on what kind of virus we're talking about. So maturity is where the viruses are going to assemble. So in release is when basically the virus is going to leave that host to infect new cells. So most of the time, like I said, during the light tick, the host cell is going to be destroyed and going to be killed. So these are the two different cycles. So again, we have attachment. That's the first, okay? So we have attachment, we have penetration, which is the virus is actually going to eject its own DNA. And then biosynthesis is where we're actually going to use the host cell is going to be tricked in order to basically the host cell is actually going to make the virus or reproduce the virus using its own DNA. So maturity is just, again, just like you're maturing, you're growing. It's going to grow the virus until it's too big and it's too large, and then that's where the release is going to happen. So the lysogenic, this is where it's going to lay dormant for a while, and again, it can actually, once it reaches a certain point, it will actually procreate or recreate, and then that's when it will be created and get released to the next cell. So again, here's just another picture. So if you look, what kind of bacteria, or excuse me, what kind of viruses up there at the top, like looks like the lunar spaceship? That's the bacteriophage. So remember, the bacteriophage injects its own DNA, and you can see the little purple. You can see that the little purple are actually the new baby viruses. So again, it just recreates so much and creates so many baby viruses that the host cell just can't hold them, and it actually explodes. So that's a light tick. The lysogenic, again, it's going to actually, if you see, the viral DNA is actually a circle. And if you notice, the green is the bacteria. And this bacteria, the green squiggly marks is kind of the DNA for the bacteria. Well, what happens is it actually fuses to the viral DNA, actually fuses into the bacterial DNA. And the bacteria, when it actually goes through cell division, it actually uses or recreates the viral DNA. So the virus really doesn't have to do much. It just lets the bacteria do its job for it. So a retrovirus is a little bit different. So a retrovirus actually, instead of having DNA, it has RNA. So the most common one, or the most one that we have research on, is the HIV virus, which causes AIDS. So instead of, again, having DNA, it has RNA instead. And this is a short little video about what we know about HIV. So, unfortunately, developing a vaccine for AIDS is really difficult because, again, it's a retrovirus. So RNA actually mutates at a faster rate than DNA. So because of it, in fact, these different viruses can exist all within the same person. And here's another short clip for you. So emerging viruses, basically illnesses that we haven't really previously known. So this is AIDS, the West Nile, SARS, Ebola, which we actually had a huge epidemic last year, and of course bird flu. So these are the ones that they have actually mutated from previously known viruses, and basically these viruses are exposed when new areas were developed, and unfortunately they can jump from species to species. So if you look at this image down here, these people are actually removing birds, so the avian flu. So they actually have, which we thought it was just associated with birds, but now we know that it can actually jump species, where it goes from a bird into us, or for the swine flu, it went from a pig into a human. So vioids these are some relatives or some cousins from viruses. So the vioids are actually way smaller than viruses, and it consists of RNA, just like a retrovirus, but it doesn't have a protein code. It doesn't have a protection. So the pyrines are actually rogue proteins. Basically, these are the ones that we think that cause mad cow disease in Kuru in cannibalistic tribes. So this is actually a protein that has gone rogue, again, or AWOL, where it hasn't done what it's supposed to do. So again, how do these vaccines work? Okay, so the vaccines, basically, once you get a vaccine, such as a chickenpox, which most of you guys probably have, your body develops this immunity to that virus, or it basically responds with an immune response. So a vaccine can be grown from either a weakened or a killed virus. Okay, most of the time we actually grow them in eggs, because eggs are actually one of the larger single cells that we can actually manipulate. So what happens is, basically, again, this form of the virus is then injected into the person's body, which causes that immune system, which causes immunity to the virus. So if you look, here's just kind of a small chart that how many vaccines or immunizations that you should have from basically birth all the way to 6 years old. So if you notice, there's a lot of vaccines that are developed or that you use or you get multiple times throughout your age. So if you look, hepatitis B, you get it at birth, 1 month, 2-month, then you skip all the way to 6, and then you continuously get it until you're 18 months. So there's a lot of vaccines, again, that you are going to have multiple vaccines for, depending on what age you are. So here's a very taboo question. So some parents have just decided, I don't want to have immunization because of some fears of vaccine safety. So a lot of the government or government agencies have actually produced commercials like the one you're going to get ready and see to encourage parents to actually get their child or their children vaccine. So after the video, during the video, I want you guys to think about, what are the risks of vaccines? Are they even safe? Would you have your own child vaccinated? And should the government actually force immunizations upon its citizens? So this is an enoid virus, and basically it's just showing you that you have the DNA that's inside, and then you also have the protein unit, which we refer to as the capsid. So here are some other images. So that's that enoid virus that you just saw. Viruses are actually really pretty. So the next couple slides, I'm just showing you guys some different types of viruses. So this is actually what the flu or influenza looks like. So again, this is another illustration to show you the difference. So this is actually, the green is the the green is actually the bacteria, and you can see the cool little bacteriophages, which are the little orange spaceship looking ones on top, that they're actually injecting their DNA. So if you look at the orange that's inside the green bacteria, that's actually the circular bacteriophage's DNA. So this is H1N1. This is the smallpox. So you have an illustration, you have an actual image, and then you also have what smallpox looks like on a child. So if we kind of compare the shape and the sizes of viruses, if you look, this pink, or the large pink cell is actually a human liver cell. So this is a eukaryotic cell. So inside that liver cell is a ribosome, which is also a dark pink color. So the bacteria is the E. coli, which is the blue. It has the pilus that's associated with it, or the little hairs. Now look at how many different viruses can actually fit inside of bacteria. So viruses are extremely small when we're talking about size. So bacteria, this is actually E. coli. So bacteria are classified into two kingdoms. It's eubacteria, which we refer to as true bacteria, and archaeobacteria, which is ancient bacteria. So they are prokaryotic, so that means they do not have a nucleus, and they're pretty much able to live anywhere, even extreme environments. So they exist everywhere. So in your eyes, in your mouth, on your skin, in your hair, in your gut, they live everywhere. So this is an illustration of kind of the basics of basic characteristics of a bacteria. So you have your flagellum, which are used for movement. Those are the tails. You have your capsule, which is very similar to a capsid in a virus. You have your chromosomes. You have your plasmid, which is a circular DNA. You have your cytoplasm. You have your cell wall for protection. And you also have your cell membrane. So the endospore is kind of like its baby spore, if you will. And the pili are there for attachment or movement. So I want you to take a minute, and I want you to look at the slide before and see if you can identify and label A through J. So what's our essential question? Why are diseases caused by bacteria? Can they be treated or cured differently than diseases caused by viruses? Well, yeah, we have to look at two things. We have to look at the anatomical and the reproductive. So when we talk about reproduction in prokaryotics or bacteria, we're talking about binary fission. So binary fission means basically you're going to split the parent cell into two daughter cells, which is very similar to cell division. But we are actually looking at asexual reproduction. So asexual reproduction means that this bacteria does not need an additional cell in order to reproduce. So bacteria actually reproduce in a couple different ways. They have conjunction, which is basically where the bacteria actually passes the DNA to a second bacteria through this tube, or it's kind of like a bridge. So the DNA in bacteria are actually called a plasmid. So they actually exchange DNA through this small bridge. The next one is transformation. This is where the bacteria takes up free pieces of DNA that's either secreted by a live bacteria or even a dead bacteria. So transduction, this is where your bacteriophages, so we remember those. The bacteriophages are those viruses that are actually going to inject themselves into bacteria. They can transfer portions of the bacteria DNA from cell to cell. It's kind of like almost like pollination, where they are taking pollen from flower to flower. So you can see the three different kinds. So transformation, you are taking small pieces of DNA. Transjunction is when somebody is actually going to, like a bacteriophage, they're actually going to add or secrete additional DNA from bacteria to bacteria. And then conjunction is that where you have two bacteria that are actually going to form that bridge, that they're going to exchange that circular plasmid or the circular DNA. So let's talk about nutrition. So we have a couple different ways that bacteria can actually eat or consume. So you have anaerobic, which we've talked about that when we talked about cellular respiration. So anaerobic means that it does not need oxygen. Aerobic means it needs oxygen. Autotroph is organisms that can make their own food. Chemotroph. Now, chemo we haven't talked about so much. So chemo means chemicals. So they actually break down chemicals in order to feed themselves. And heterotroph is where you have to eat other things. So mutualistic. We've talked about mutualism when we talked about ecology. So mutualistic means that they need a partnership. So when we talked about mutualistic nitrogen-fixing bacteria that live on roots of plants, they actually are reducing nitrogen to ammonia that we can actually use. So we also have mutualistic bacteria that live inside of our intestines. So basically those plant products that we just can't consume and we can't just break down, that's that bacteria that's going to assist us with it. So your appendix. Your appendix is basically thought to be kind of like a reservoir for good bacteria. And basically that's what we thought. Remember, it is a vestigial organ, so you can have it removed and it's not going to hinder you whatsoever. But that's the one idea or one theory that we have for the purpose. So we think that it's basically where the good bacteria is being housed. And some scientists actually think that that is where the bacteria that breaks down cellulose or plant sugar that we can't just break down is housed as well. So there's a couple different shapes. So you've probably heard of strep, right? Strep throat. Well, what the heck does strep actually mean? Well, streptococcus is actually strep throat. So strep means in chains. So if it is strep, it's in chains. If it's coccix, coccus, that means that it's spheres. So streptococcus, for strep throat, means that the bacteria is circular and it's in chains. So bacillus is rods and spirilla is spiral. Well, a staph infection, instead of being in chains, it's actually in clusters. Kind of looks like grapes. So here is just a small diagram to show you the different types of bacteria that are common through us. So E. coli, streptococcus, staphylococcus. Okay. So this is streptococcus. So again, strep means what? In chains or in clusters. Strep means in clusters. Coccus means circular. So gram stain. This is actually, remember how we talked about dyeing, where we had to actually dye our cheek cells? Well, gram stains is basically a procedure that we can actually look at to identify some bacteria. So if it's positive, that means that it has a peptide glycogen, that actually traps the violet. If it's negative, that means that it does not have a peptidoglycan cell wall. So this is some negative, which are pink. This is positive, which they are purple. So what I want you to make sure that when you're going through your bacteria lab is how are they grown and how are they analyzed. So make sure that you brainstorm how are bacteria actually grown and then how are they analyzed and identified and named and studied. So I want you to identify this one. It is Bacillus. It is gram negative. So what is this one? This should be Staphylococcus. Staph in clusters, coccus circular. And it's positive because it's purple. So this is Streptococcus. This is, again, responsible for strep throat and also for flesh-eating bacteria. So if you can see, staph means bunch of grapes in Greek. So at the person to the top right, you can see that this is actually eating that person's flesh. So Bacillus, this is gram positive. This is actually a really bad example of a stain because it kind of looks purple and it kind of looks pink. So this is actually really easy to grow in the lab and it's not very harmful to us. So we like to use this in lab activities. So this is E. coli. This is what is associated in your gut and helps you digest some of your food products. Salmonella is associated with chickens and holding turtles. So this is just a really cute slide because I thought that, and you can actually purchase these plush germs. So you have syphilis, you have E. coli, you have gonorrhea, and then you have the plague. So the germ theory. Germ theory started around 1860. So basically this is the single most important contribution that science has ever had when we're talking about microbiology. And basically before 1860 we never washed our hands. So you might have been a surgeon and you go from patient to patient and never washing your hands and never washing your tools. So bloodborne pathogens were being spread among all of those patients. So this theory is actually, before, again, we thought that it was voodoo. We thought that it was taboo. We thought it was magic, that why you got sick. Well, we now know that the reason why you catch diseases, or some cause of diseases, not all, are due to those microorganisms like viruses and bacteria. So the key to developing the germ theory of disease was actually, again, a response or a rejection of spontaneous generation, which is, again, we thought magic, with things just magically appeared out of thin air. So now we know, with that, we know that microorganisms do cause some, or if not all, diseases, and that's where we have some methods of prevention, such as spoiling of food through pasteurization, and also just basically washing our hands and our bodies. So why is this a theory and not a fact? Because can we say that all diseases are caused by microorganisms? Unfortunately, we cannot. So the three different gentlemen, or three different scientists that are the contributing factors to creating the germ theory, are Pasteur, Notch, and Fleming. So Pasteur actually developed or helped develop the germ theory and disproved spontaneous generation in 1885 when he actually developed the rabies vaccine. Notch's postulates, which we're going to talk about when we get to our graphic organizer, are the statements that where he actually found that microorganisms did in fact cause most, or not if all, diseases. And Fleming is our number one vaccinator. He actually discovered penicillin, and we actually use this. This is our number one antibiotic that we use every day.